everyone. My name is Joy, and I'm a second-year grad student at Cornell in the Applied Math department. I just actually switched from math to biology, ComBio, so this is one of my first projects. So today I'll be talking to you about the geometric analysis of admixed taxa in the neighborhood joining algorithm. These are my collaborators. Okay, so distance methods are a popular way to infer phylogenetic trees from genomic sequence data. A neighbor joining algorithm is a type of distance-based methods. So what neighbor joining algorithm does is it's a greedy algorithm in every iteration. So in every iteration, it tries to pick the closest pair of taxa. And I'll define how this algorithm defines closeness. So the neighbor joining algorithm measures closeness between a pair of taxa by this thing called Q criterion. So the idea is to minimize the distance within the pair of taxa picked in every iteration and maximize the distances between that pair to every other taxa in the tree. So every time a pair is chosen, we'll remove that pair of taxa from the tree and add a new node. We call this removal and addition a cherry. And the new distance matrix will be updated, and the new distance matrix will involve one fewer taxon, thereby having one fewer row and one fewer column. And then this is how we update the distance matrix. So what is admixture? So admixture happens when two populations interbreed to produce the third population. This phenomenon can be empirically observed, as shown in an example, where we have an admixture between Native American population and European population. When we use the neighbor joining algorithm to infer trees of population with admixture, we need to account for this additional structure because it changes the input distance matrix. It basically creates an additional structure in the input distance matrix. Currently, we don't know how to account for this extra dependence relationship among the entries of the input distance matrix in the vanilla neighbor joining algorithm, and we also don't know if the tree inferred by the neighbor joining algorithm is reliable when there's admixture behavior. Therefore, this is the motivation for our project to study the feature of to study how to have admixture in the neighbor joining algorithm. So in this model, we're going to assume that the distance function is linear when we apply the function to admixed populations. So the distance between an admixed population and all the other populations can be expressed as a linear combination of the distances of its source populations. The coefficient alpha represents the proportional impact of the source taxa on the admixed taxon. So in the picture, the taxon colored in red are the source populations, and a taxon colored in blue is the admixed population. And the matrix D is how we construct the distance matrix as the input of the neighbor joining algorithm when there is admixture among the populations being studied. In previous work, Koppelman et al. Have proved for n equals 4, there are 3 properties that usually happen when the data of the neighbor joining algorithm has admixture. So these are the 3 properties. And they showed it for n equals 4 and some very specific type of input data. So in our project, we're going to extend this for greater n. In particular, in this talk, I'm going to talk about n equals 5 and also view it from the perspective of combinatorics because I'm more familiar with that method than other methods at the beginning of my PhD. So what is the question that we are concerned about? So the question is, what is the probability that an arbitrary neighbor joining tree inferred from input distance matrix with admixture satisfies the 3 properties that we just talked about? So what are those 3 properties? So first, we have antecedents. So there are 3 ways we can cluster the taxa together in the neighbor joining algorithm, and so these are listed as A, B, and C. Only A and B satisfy property 1, meaning that only in A and B we have that one of the source taxa either 1 or 2 is clustered with the admixed taxa N before the source taxa 1 and 2 are clustered among themselves. So only the first 2 options satisfy property 1. And in property 2, we say that this property is satisfied if and only if the distance between the 2 source taxa is further apart than a distance between each of the source taxa and an admixed population. And in this case, we see that the distance between the 2 source taxa, 2 and 1, is 1.4. The distance between the one of the source taxa, 1 and 5, the admixed taxon, is 1. And similarly, the distance between taxa 2 and 5 is 1.2. So the distance between the 2 source taxa is indeed the largest. So this property is satisfied. Similarly, for property 3, it states that the number of edges, so we don't ignore the values on the edges, we just focus on the number of edges between 2 nodes. The number of edges between 1 and 2 is 4, which is greater than the number of edges between 5 and 1 and 5 and 2. Therefore, property 3 is satisfied in this example. So we want the number of edges between source taxa to be greater than the number of edges between source and admixed. Okay, so in our method, we don't want to deal with distance matrices. We want to deal with distance vectors. So we will be flattening the lower triangle of the distance matrix into a distance vector. Because the distance matrix is a symmetric matrix, the lower half is the same as the upper half, and we will flatten the lower half in the reverse lexicographic order in order to get the vector. So our method wants to eventually interpret the neighbor join algorithm as polyhedra, which was first introduced by Yoshida and Eichheimer in 2008, and our method will build upon the theory they developed back then. So in order to do that, we know the polyhedra are defined by hyperplanes, and then hyperplanes are in turn defined by inequalities. So then so the goal is to first turn the neighbor joining algorithm into a bunch of linear transformations, so then we get these linear hyperplanes. So first, I'll explain why the Q criterion can be interpreted as a linear transformation. Recall that we have this Q criterion from a previous slide. It has been proven that there exists a matrix A that can transform the input distance matrix or vector into this Q vector. Yes, through linear transformation, and this is an example when n is equal to 5. Similarly, even the distance update process can be interpreted as a linear transformation. And in this example, when n is equal to 5, and the cherry that we pick is taxa 3 and 4, we have R written this way. So this step of the algorithm is also linear transformation. Okay, now we know that in every iteration of the neighbor joining algorithm, we want to pick the taxa that corresponds to the entry of the Q vector that has the smallest value. So this is kind of like adding restrictions to a pair of taxa. And those restrictions, because of the fact that we can turn every step of the algorithm into linear transformations, those restrictions themselves are also linear conditions. So we can use those linear conditions to get these linear inequalities that we will use to construct hyperplanes. Okay, so just a very general overview that this is a picture from this book called Polytope, and basically it's just to show that we can go from inequalities to cones, which is a type of polyhedra. So far, we have explained that we can go from the neighbor joining algorithm to inequalities. And from inequalities, we can get hyperplanes. And from hyperplanes, we can get these cones. And then the theory basically states that there's exactly one cone that corresponds to one inferred tree topology, not up to a reflection. So we now have a pipeline from the cherry-picking order from the algorithm to exactly one tree topology that corresponds to a cone. Where are these cones? These cones lie in the space of distance vectors. Two distinct distance vectors in the input distance vector space can correspond to the same cherry-picking order, which means that if 2 points lie in the same cone inside the input distance vector space, that means that those 2 points, after we apply the neighbor tuning algorithm to them, we will get the same cherry-picking order as the output. So, and we also know that every input has some type of cherry-picking order as the output. So every single point lies in some cone in the space. So these cones completely partition the space of input distance vectors. So far this is a method for any distance inputs, but we want to focus on the subset of distance inputs that satisfy this admixture behavior. So how do we do that? We want to embed this smaller space of admixed distance vectors into the larger space of all distance vectors. Okay. So we do this by adding an additional taxon to the admixed distance vectors, and then we can embed the longer version of the vector into the distance space where we can apply the method I just discussed. Okay, so how do we test whether a vector is contained in a cone or not? We do this multiplication of the cone's hyperplane conditions with the embedded vector. And if it's less than or equal to 0, then this embedded vector is contained in the cone. And after that, we will basically compute which cones in the space satisfy property 1 and which cones satisfy property 3. And then we'll use this count, or more theoretically, the volume of those cones relative to the other cones' volumes to compute the probability that an arbitrary distance input could generate a tree possessing one of the 2 properties. So this is an example where a cone does not satisfy property 3. And these are all the cones that don't satisfy property 3. Okay, and property 2 is more complex, where we will only a subset of the cone can satisfy property 2. So it's not just a yes or no question of the cones. It's more like we need to subpartition the cones to see which points satisfy the property or not. So so property 2 is more complex. So the way that we go about this more complex issue is first, do another linear transformation of the input distance vector to the output distance vector, and then finally we want to do the comparison to get property 2. Okay, so I can skip to the results then. So using this method of computing cone volumes, we were able to get results that match previous simulation-based empirical work on this topic up to a pretty good precision. So all we did was decide which input distance vector lies in which cone, and then we compute the volume of the cones that satisfy the properties that we are interested in. And this is a theoretical method that doesn't rely on simulation and empirical experiments, and somehow our results match pretty closely to the empirical observations from previous work. So yeah, our method provides a theoretical, more principled way to study this topic, and I guess it's another perspective to understanding a neighbor tuning or algorithm. Okay, and this is my lab, and also the mathematician that I work with at Cornell. Yeah, thank you.
It's 10 on my clock. So, okay, our next speaker is, he just told me how to say
It's not just you. Many people cannot pronounce my name. Xianfa Xie.
Xianfa Xie, thanks. And he's going to talk about why have most of the phylogenetic studies of COVID virus evolution been wrong. All right, thanks.
I'm looking for Scott Edwards. It's see if he's here, because he told me my title is too inflammatory. All right. Again, my name is Xianfa Xie. I'm working at Virginia State University, the center, director for the Center for Biotechnology, Genomics, and Bioinformatics. First, I'll plug in here. It's a new center. We're hiring people. If you're interested in any of this field, please let me know, and we can connect with each other, a meeting, and also working at Department of Biology. So one thing I have learned so far in my career is that you cannot be too radical. So that's why Scott told me that your title is so inflammatory. Um, but, I mean, just give you an example. 2009, I organized a symposium on epigenetic environment and evolution. I believe that's the first symposium in evolution committee on epigenetics. At that time, not many people were thinking about epigenetics, but now I get, particularly at this meeting, I saw so many talks on epigenetics. So that gave me hope that sometimes if you try to advocate something, so that may have some effect. All right, so back to my talk. So I'm looking at virus evolution, particularly this SARS-CoV-2 that's called COVID-19. Everybody have seen this image before. And this is the genome sequence of the coronavirus once it has. Okay, thank you. And again, first discovered in November 2019. The genome sequence was quickly announced and then compared with some other closely related virus. Since then, of course, many variants have been discovered, many lineages have been identified, and these are two most important systems characterizing or classify the different lineages of virus. So GIS-AID system and Pango system, both of them, as you notice here, they use a phylogenetic tree approach. Many other studies about the SARS virus evolution has also been using phylogenetic tree approach. But you probably have noticed something very interesting out here. Oh, sorry. Is the pointer here? Top, okay. Right? As you can see here, there are some very long branches, and everybody familiar with the phylogenetic tree construct, you know that this is the indication of something going on, probably recombination happening. All right, so this is again the lineage found in Brazil, right? And even more weird, some other studies showing here there's a very common used website, a database for tracking different variants of virus. As you can see here, they are looking at Omicron. Just look at where it, using phylogenetic tree, just look at where it goes back to. Early 2020. So what does it tell us? I mean, do you believe that's true? If it had happened, it could be traced back that very early on in the pandemic, and nobody has been able to detect any of the sequences along the way. So that, to me, just unbelievable. So again, this long branch indicates something else going on. Again, recombination. And there are some other signs of recombination in some other variants, including alpha, beta, delta, so on. So as you can see, also indicated by these long branches leading to some of the major variants. All right, so the signs seem to be clear there, but nobody was giving any attention to recombination. But recombination has happened in virus evolution. It happened in HIV virus evolution, in some other coronavirus evolution. But for all these studies, the community or some of the major groups working on this SARS coronavirus evolution had not been paying much attention to the role played by recombination. And again, mostly because they have been using phylogenetic tree, which cannot accommodate, is not a very convenient way to study recombination. Again, there are some other signs of recombination out there. So just look at the table. Look at all the recurrent mutations that happen in different variants. Again, some people suggest that it's just, mutation that happened independently at the same site. But I would think it's more likely to be the result of recombination. Okay. So again, as I mentioned, most studies so far, published studies so far have been using phylogenetic tree, but as we all know, when you do a phylogenetic tree, you need to check all your assumptions, or any population genetic, including population genetic method, you need to check your assumption, or phylogenetic tree analysis, you need to check assumption. There are many assumptions with phylogenetic tree analysis. So the first one, of course, is all ancestral species or sequences have gone extinct, right? So all the species you are studying now should be put at the tip of the tree. All right. This only possible if all the ancestral species or sequences have gone deep, or not included in your study set, in your data set at least. But in the case of SARS coronavirus 2 evolution, you might be looking at both ancestral sequences and the descendant lineage in the same data set. All right? In that case, actually they have this ancestor to descendant relationship. You kind of put all of them at the tip of the tree. The other thing about the phylogenetic tree, ideally you want to make it a bifurcating. So if you have multi-bifurcation, that is you would consider that's not a very well resolved tree, all right? But in the case of ongoing SARS coronavirus evolution, so you could have one ancestral virus lineage giving rise to many different descendant lineages, all right? And following the tree, if you want to get a very clear signal, a well-resolved tree, so you need to have a deep divergence with fixed differences among species or among the sequences you are looking at, right? But in the case of SARS coronavirus 2 evolution, ongoing evolution, so those sequences are maybe very similar to each other, all right? It's hard to find a very clear signal among them if you are using phylogenetic tree analysis. Again, last but not least, everybody know that. So to use phylogenetic tree analysis, you need to assume vertical or linear inheritance, right? So no reticulate evolution, including recombination, can be accommodated easily by phylogenetic tree analysis, right? But again, as I just mentioned earlier, so recombination, given all those evidence I showed in the previous slide, seem to be there, seem to have played a very important role in the evolution of the SARS coronavirus 2 virus, and particularly in the production of those major variants, right? So that's why instead of phylogenetic tree, I would suggest that I would advocate for phylogenetic network approach, right? So in contrast to phylogenetic tree, phylogenetic network approach, what's the difference, right? First, instead of assuming vertical linear inheritance, phylogenetic network can accommodate non-vertical inheritance. That include lateral gene transfer, hybridization, recombination, okay? And instead of assuming distinct and extinct ancestral phylogenetic tree, you can have both ancestral sequences and descendant lineages within the same data set, right? And recent ancestors, that's fine. That can be studied in phylogenetic tree network, okay? Again, phylogenetic tree requires significant fixed divergence among species among the sequences you are using, but phylogenetic network can accommodate divergence at any level, all right, including within populations among the different lineages of viruses. Bifurcation, again, that's ideal for phylogenetic tree, okay? But network, you don't need that, all right? You can have multiplications. One ancestor can give rise to very different descendant lineages. So if we think about a phylogenetic tree, sometimes actually we are force everything into a tree, right? It's a simplification for, in many cases, it may be just a simplification for our data set. So phylogenetic tree, I would argue that is more realistic and inclusive because you do not need to force everything into a tree, into a bifurcating tree, all right? So just let the data to show how they relate with each other, okay? But of course, there are some challenges about phylogenetic tree, phylogenetic network approach. Again, the tree method have been well developed, as you have seen in this session. All right, they have people are still developing or refining different methods to construct the phylogenetic tree. All right, but phylogenetic network, of course, is much more complicated. Magnitudes are more complicated than phylogenetic tree construction. There are some other challenges that are shown here. So the results may be less appealing than phylogenetic tree. Phylogenetic tree is so beautiful, you can so clear, and you can, describe the phylogenetic tree relationship in many different ways, but phylogenetic network seems to be very messy, as you can see here. And it's hard to interpret, and the algorithm involved to build a phylogenetic network, of course, is much more complex and computationally more costly, okay? And so far, there have been much fewer software or methods developed for phylogenetic network analysis compared to phylogenetic tree analysis. And of course, a major hurdle, I believe, is that right now in our community, we still think phylogenetic tree should be the dominant method to use, all right? And have been accepted as the dominant method to use. But again, I would argue that we should move from phylogenetic tree thinking to a network thinking, okay? And again, because it accommodates many other situations. So we did some preliminary study using phylogenetic network to analyze the SARS coronavirus 2 evolution. But our study was not the first one. There was actually previous phylogenetic network analysis published in PNS, but, okay, I need to hurry up. But that one had a chilling effect. After that publication, no publication of phylogenetic network analysis of coronavirus to ever been published. So we will look at alpha variant again, just look at the recurrent mutations that happen in the coding ring, particularly in the spike protein coding region. You find some recurrent mutations between different lineages of alpha variant that suggest, again, recombination. Again, some other signal of recombination, long branches leading to alpha variant, okay? And we did a phylogenetic tree analysis, weak support, okay? Then we used some phylogenetic network approach to analyze the data. So as you can see, we tried two different methods, and here, that's the alpha variant, okay? As you could see, it could be traced back to the combination of two different lineages, one lineage and the other lineage combined together to give rise to the alpha variant, okay? So this is one method we tried. And the other method is, I call this the satellite plot. It looks like satellite, so also look at that sequence here, that's alpha variant, okay? So again, it seems to have come from recombination of different sequences that exist in the population, all right? So we made this public in in June 2021 on bioRxiv. If you're interested, you can check it out. So at that time, it was not accepted. Again, we submitted our manuscript to PNAS. As I mentioned, no, rejection, okay? The committee didn't accept it. But since then, there have been more recombinant sequences recognized for the SARS coronavirus, including a paper published by same group of people who previously used phylogenetic tree to study Omicron evolution. Now, even though they're still using phylogenetic tree here, but they recognize there may be recombination, okay? So again, to summarize and to conclude, I would just say that the phylogenetic network compared to phylogenetic tree may be more inclusive, and I would encourage people who are developing phylogenetic tree method now to spend maybe a little bit more effort to better develop phylogenetic network method. Thank you very much for that.
Okay. Hi, I'm Jonas Lescroart. I'm a last year PhD student at the University of Antwerp with Prof. Hannes Vardal and the Pontifical Catholic University of Rio Grande do Sul in the south of Brazil with Professor Eduardo Ezerick. And I'll talk about cats and concordance factors. So these are the cat species that I work on. They're all domestic cat-sized. They occur in the neotropics. I won't talk about them much during the presentation, but I didn't want to withhold these beautiful pictures from you. But please come up to me afterwards, and if you want to talk about the biology of these really fascinating group of animals. I'm a bioinformatician mostly, so I don't really go into the field. This picture with me was in a rescue center. But I definitely want to thank all the field biologists who collected our samples And many of them are associated with these grassroots, bottom-up NGOs all over South and Central America. So thank you to all of these people. Quickly about my data, I use whole genome short reads. We have a normal mammalian genome size. I have for this study 15 genomes, and we use 2 different very high quality reference genomes, one in-group and one out-group species. And I'm using SnakeMake as a workflow manager, and my code is also available on GitHub. So the main question of my PhD is about the phylogeny and the taxonomy of this group, because it has been controversial since forever, and I wanted to do something about that. So what does the phylogeny look like? How many species are there really? And here's the answer, kind of. Not really, actually. On the left, you're looking at a whole genome neighbor joining tree, annotated with bootstrap support. And on the right, you're looking at an astral species tree with local posterior probability support. And you'll see that the ocelot is in a different position in these phylogenies, even though support is at its maximum at every node. And so we iterated over a bunch of different combinations of our data. So we used the 2 different reference genomes for all analysis. We in or excluded repetitive content in our analysis, and we tried different methods for local tree reconstruction, so both neighbor joining and maximum likelihood, and then different summary methods. And we see that sometimes one of the taxa jumps around, even though support is uniformly high. So what's going on? And the answer is, of course, gene tree heterogeneity. And to have a look at the topological variation along the genome, what we would typically do is we take the genome, the red bar, and we break it up into fragments. And these fragments go by many names in the literature: genomic fragments, windows, loci, haplotypes, C genes, and I'm sure there are others. In my case, I used 100 kb non-overlapping windows or genomic fragments. And then from each fragment, we do a local tree inference. So I used neighbor joining and maximum likelihood. And then these resulting trees or set of trees, we call these local trees, locus trees, meiotic trees, or gene trees. And from that set of trees, we make a summary, a consensus tree, and we annotate it with concordance factors to have an idea of how much support each node finds throughout the genome. I do want to make a plea here to please stop using gene trees, because only a small fraction of the genome is actually in genes. We're doing a whole genome analysis, so most of our trees are not at a gene, and yet they're very often called gene trees. So can we please stop calling them gene trees? I prefer local trees because you can oppose it to the global tree, which would be the consensus tree. I've also seen this meiotic trees, which I think is quite nice, but I suspect it's very little known. It's derived from mereology, the study of part-whole relationships, but maybe that one will be a bit hard to catch on. And here's an example from my data. So this is a X chromosome of an alignment of 5 leopardus species, and I'm sure you've seen these barcode plots before. Every color represents a different topology. So I'm using 100 kb fragments, and there's certain assumptions about recombination within and between fragments. I know about this, but I think we shouldn't be too worried, because there's still, as you can see, plenty of resolution to detect alternative topologies. Right, so how do we summarize this? One way visually is to overlay them on top of each other. This is a density tree plot. We can clarify that a bit by showing only the most frequent crosslinks. This is an implicit phylogenetic network. And if we summarize that even more, we come to a phylogenetic bifurcating tree annotated with concordance factors. So the first number is the concordance factor, and the second is the bootstrap support. And the concordance factors that I use are as defined originally being the proportion of the genome for which a clade is true, and a clade being a monophyletic grouping of samples. So the hierarchy within the clade is not important. It's as long as you have a set of samples, then that defines the clade. So to interpret this, the top one means in 99% of the genome, we find these two eastern tiger cat samples together. And I've calculated this with some trees. Now here's the main message of my presentation, which is that concordance factors are fundamentally different from statistical measures of support like bootstrap or posterior probability. I'm definitely not the first one to say this or to point this out. From its very inception, it's been said that if the concordance tree were annotated with concordance factors on nodes, these should not be confused with measures of support. And to phylogeneticists, this may be trivial, but my impression is that in the wider biological community, this is not realized as much as it should be, and I'm not the only person to think so, because earlier this year we had there was a review preprint, the meaning and measure of concordance factors in phylogenomics by Lanfear and Han. I highly recommend this paper. Okay, so concordance factors themselves and discordance factors, their complement, these are estimators of a biological property, which is the amount of topological variation along the genome. And to illustrate this, let's say we have two topologies in the genome, and they occur at some true frequency. And we estimate this from the data, and then we have a confidence interval for these concordance factors that we estimate. And as we add more data, our precision of the concordance factors will increase. But the concordance factors will not go up like a statistical support would go up. So this is very different from statistical measures of node support, which are a measure of statistical confidence. So given our data, how likely are we to consistently retrieve the same result? And pretty much with this, if we throw more data at it, it'll go up and up until eventually it's at a plateau, right? So this is a back of the envelope simulation here, showing that with the increasing number of data points, you'll go to a maximum statistical support. Even if, in this case, and it's moving about a bit, which is weird because I used PDFs, but okay. So in this case, I had two topologies, one at 51% of the genome and the other one at 49% of the genome. And still, if you have enough data, you'll get to nodes with 100% bootstrap support, which is the case, of course, in modern phylogenomic data sets. So that makes these bootstrap supports pretty much useless in some cases. So here's my results for the Leopardus species, and we'll break it down. So first thing we identify are some nodes with full or 99% concordance, and these are the species level nodes. So for some species we had multiple individuals. And then for two of the species, the ocelot and clouded tiger cat, we had 3 samples, and you'll see that within the species there's low concordance factors, so 46 and 54 there, and those represent intraspecific gene flow, so gene flow between populations. That makes sense, but then the species as a whole is monophyletic throughout the genome. Oh, and I should say that the clouded tiger cat is a new species, actually, so you've probably never heard of it. Right, and then we have a bunch of other nodes with low concordance, and there the assumption is that by default we would blame incomplete lineage sorting, and then we do introgression tests to see if introgression also contributes to the low concordance. So that's what we did, and I've used 3 different tests for introgression, one based on site patterns with D-suites, one based on topologies with phylonetworks, networks, and then on distribution of branch lengths throughout the genome with Quibble. And they show the same a consistent result, which is introgression between the southern tiger cat and the Joffrey's cat, which we actually knew about from an ongoing hybrid zone in the south of Brazil, and introgression between ocelots and this subgenus Oncyphelis. And then the remaining low concordance in the tree we can blame incomplete lineage sorting, which is actually consistent with the history of 2 waves of quick successive speciations in this genus. The oldest one being in the early Pliocene in Central America, and then the more recent one endemic to South America in the early Pleistocene. So thank you for listening. I have a poster this evening as well, so you can come up to me and talk about this. I'm also looking for postdoc opportunities, but specifically we're in Europe because my girlfriend is coming with me. And this presentation was based on this paper here at the bottom. So thank you very much. Oh, if there are questions, I'm happy to take them. Yes.
How old is this clade in part 2? Are you finding really short internodes, I guess, for the poorly supported nodes?
Right, so the question is about the age of the clade and the length of the branches. So the age of this clade, we estimated the base at 5 million years ago. And we see that some of the, well, this is not a time-calibrated tree, but some of the branches are really short, and those are the waves of quick speciations that I'm referring to. And they have a lot of incomplete lineage sorting, and therefore also low concordance. Yes?
Okay, and for the last talk in this specific section, we have Peiyu Xu, which will introduce a new simulation tool for epidemiology that includes ecoevolutionary factors as well.
Okay, cool. Hi everyone, I'm Perry, and I'm a second-year PhD student in Genetics, Genomics, and Development Department in Cornell University, and I'm supervised by Dr. Jae-hee Kim and Dr. Andy Clark. So today I'm really honored to be sharing with you about my research. Today what I'm going to introduce is a novel epidemiological, ecological, and evolutionary simulation framework for genomic epidemiology, and the tool's name is called E3SIM, because there are 3 E's. And before I'm going to talk about what my simulator is, I'm going to talk about why a genomic epidemiological simulator is important. What can we know about the genetic data of the pathogens? So the pathogens' genomic data are a result of a genealogy of these pathogens, and these genealogy are a result of the pathogen epidemiological dynamics. If we think about this process forward in time, we were representing these gray circles as the host. When a host is infecting another host, that will be shown as a branching event on the genealogy of the pathogen, because the pathogen in the original host body will plant an offspring of this pathogen into another host body. So that will be shown as a birth event on the genealogical and the genealogy. And when this host is being sampled or the host is removed from this epidemic, it will be shown as a termination event, shown by this red cross. So all these epidemiological events are recorded on the tree, and from the genetic data where we sampled at the termination points from the tree, we will be able to infer all these events backward in time, and we could be able to infer the birth rate and death rate of these processes. So for example, if a pathogen is having a higher transmissibility, you will often see a very high branching rate on this tree. Here I'm showing a screenshot from NextString, a pathogen evolution tracking database, where you can see that this is data from COVID, and you can see that there's a strain called JN1 that is being very popular recently, and you can see there's a very high branching rate on this subtree, and also this strain in laboratory is being shown to have a higher transmissibility, so that align with the pattern we see on the phylogenetic tree. So there's numerous biodynamics methods being proposed to infer all these epidemiological parameters from the tree, including the transmissibility we just talked about, and often also a lot of other parameters. Some of these methods are model-based, some of these are not. And the thing is that you do not know which method is the best one, and sometimes what they infer are just they're just different between methods. And how do we know which result is correct? That's why we need a simulator. To validate all these inference methods, we need synthetic genomic data with ground truth parameters, because in the real world, you never know the real parameters. And also, these ground truth parameters and simulated genomic sequences are very important for the machine learning methods, for example, Phylodeeps, to train a model. So, and there are actually a lot of existing genomic epidemiological simulators, but why do you want to design a new one? There are three main aspects I'm going to introduce that our simulator has this feature, but some other simulators do not. The first one is that existing simulators, they often decouple evolutionary process and epidemiological process. What I mean is that, so if you see this scheme, which is a very popular simulation scheme for most of the genomic epidemiological simulator. So first of all, they have this population dynamics, for example, the SDIR compartmental model, and also the host contact network. And from that, they generate a genealogy of the pathogens solely based on this population dynamics. And then they superimpose mutations on top of this tree based on Poisson process or any model you want, and then they got the data of the sequencer at the end of the at the tip of the tree. But what is the problem of this process? So it's assuming an independence between this tree generation process and the mutations, which is not correct in the real world. So in the real world, when the tree is when the transmission process is proceeding, when the mutations happen on some lineages that confers a higher fitness, for example, a higher transmissibility, then the tree shape is going to be changed. The offspring of that specific lineage is going to branch more through this process. So the data structure you will see, for example, the frequency of the alleles will be very different from the data you will get from this independent hypothesis. So that's one aspect we want to improve in our simulator. And the second reason is that a lot of existing simulators, they often do not incorporate a wide range of ecological complexities. For example, there are pathogen site complexities, like the pathogen, they do not only branch out when they infect others, they also branch out within hosts. So the tree you'll see is not only a result from the transmission process, also the within host reproduction process. And we also have superinfection events where the one host could be infected with not only one lineage of pathogens, but a lot of others. And there could be latency, like when the host is first infected with the pathogen, it's not infective. So, but the pathogens still evolve in the host, but the host cannot infect any others. And there are also host side properties. For example, the host contact network is not homogeneous. Like every individual host has their own contact network, and they can only transmit the disease to someone they are in contact with. Where I'm showing two different structures than the homogeneous network. And the first one is the super spreader, where like there are some hosts that are very socially active, so that when they are infected with a pathogen, they are more prone to infect others, and there are subpopulation structure, where, for example, in a region, we could have urban and other rural areas, and the contact rate within those areas will be very different. And the third reason is that a lot of existing simulators are really hard to use, especially for people who prefer viral interactions. Here I'm showing a screenshot from another very cool simulator called Centasim. And you can see that to incorporate evolutionary compartments, you have to write your own code, write your own operator, which is kind of intimidating for people who do not want to write code. So we try to incorporate all these aspects in our new simulator. The first one, we coupled evolutionary process and epidemiological process by using SLIM as backend in our simulation. So we're simulating the epidemiological process and the evolutionary process at the same time, and they interact with each other. And we incorporate a wide range of ecological complexities that we just introduced. For example, we explicitly model the host contact network, and the transmission can only happen between the nodes that are in contact with each other. And we also design a GUI, a graphical user interface, so that any people can just use it without being obligated to write some codes. And then finally, I'm going to talk about the simulator. So before conducting real simulation, there are some pre-simulation modules we provided to generate the whole simulation configuration. We provided 4 modules. In the first module, we generate a host contact network, which based on some random graph model, or users can also input their own contact network based on the real data they want to study. And we have many like very simple or classic network models, for example, Earls-Rennie graph or Barabasi-Albert graph. And in the second module, we could generate some initial pathogen genomes, which are kind of the seeds in this outbreak, and we could use the Wright-Fisher model or just producing some sequences using a very small outbreak simulation on top of this network we just generated. And we'll have these seeding sequences with different mutation profiles and also their genealogy, which is ready for seeding the true simulation. And the genetic architecture, we could, wow, what are those squares? But yeah, okay. Anyway, so we could generate, you could define, or we could generate a genetic architecture for you that would define the effect sizes of all the mutations you have in the genome, so that every genome will have their own trait values. The important traits such as transmissibility or drug resistance will be different for each genome if they have different mutation profiles. And then we can seed the host, and we provide some algorithm to seed the host. For example, you might want to seed a very low connectivity host with a very high transmissibility strength, and those can all be done in this pre-simulation configuration. And then there's the real simulation. It's a very easy to understand process. So we have this contact network, and we infect the patient zeros with the seeding sequences. And then they can only infect the hosts that are in contact with infected hosts. And then during this process, we record this genealogy of the pathogens, and the mutations happen simultaneously with this process. And then the mutations could change the trait values of this these pathogen genomes based on this additive genetic architecture, which we have already defined in our pre-simulation modules. So every mutation will have effect size, and the trait values will be sum of all these effects of all the mutations. And here I'm showing the trait is drug resistance. So they will affect the recovery rate or treatment successful rate of these pathogens. And for the host side, there are going to be 4 compartments: susceptible, exposed, infected, and recovered for all the hosts. So, and all these transition rate between these compartments are, they could have a time-dependent feature, so that you could implement different stages of this, different stages of the outbreak. And also, we have this graphical user interface for people that do not want to interact with coding, so that you could conduct everything just by clicking on this GUI. And this is helped by 3 very talented undergrads and research assistants in our lab. And I'm going to show some examples. Ooh, the plot does not look like what I uploaded, but anyway, what you see in the left is SEIR trajectory, and I have 10 replicas there, but obviously you can only see 1 line. But anyway, that's the number of hosts that is in the different compartments: susceptible, exposed, infected, and recovered. You can see that we impose 2 different treatment stages for the whole population sequentially. And when the treatment is imposed, you can see that the whole dynamic got changed, and infected individuals got decreased. However, they kind of surged back afterwards, and most of the pathogen lineages after this treatment will be having the resistance to these treatments, like you're going to see in the right graph, where the resistance are shown by the heat map in the bottom, and the phylogenetic tree is colored by transmissibility. So you can also see that during this process, the overall transmissibility of the pathogen population got increased. So this example kind of shows that there are going to be the emergence of many new strains that have new traits, for example, resistance or high transmissibility, which is kind of affected by the selection pressure we have on this population. And we're going to show another application when we are simulating Mycobacterium tuberculosis transmission dynamics in a specific region, Karonga, Malawi. And in this example, I'm going to show the impact of using different contact network when you do simulations. So we're going to compare our simulated results to the real data set. So first we have MTB sequences from the real world, and then we reconstructed maximum likelihood tree using IQ-tree. And then we infer their effective population size trajectory in the history using a package called Phylodene. And then, cool. So this is the real data trajectory. And you can also see this bottom heat map is the coalescent frequency. And Then we use the Erdős-Rényi graph, which is a graph that every host is having the same expected number of connections. So the contact degree distribution will be kind of anomaly distributed. And using this contact network, we simulate some MTB outbreak, and we sample sequences from them and go through the same process, and we draw this plot. And we can see that this Erdős-Rényi graph simulation kind of really replicates the real data very well, having very similar past NE history. However, if we use the Barabasi-Albert graph, which is a graph that's super spreaders in it, so some of the hosts are very highly connected with others, whilst the others are not, and go through the same process, we see that it's not that well aligned to the real data set. So this just shows that contact network is a very important property to incorporate when you want to simulate the real world transmission. So with that, I want to thank my both lab members, and this work is a collaborative effort of these people in my lab, and also special thanks to our collaborators who provide the data and advice in the designing the simulator. And we have this manuscript on bioRxiv. Please check it out. Thank you so much, and I'm ready for any question. Yes, please.
So how do you accommodate the situation where the same individual may be infected by multiple strains of the virus? The possibility of recombination. Can you
Yes, as you see, like first of all, our simulator do not incorporate recombination. That's kind of a not good thing. We're going to do that later because in Slim we could readily do that. And then for multiple strains of the same individuals, because all the pathogens they could transmit to the other individuals, if you turn on the superinfection function in our simulation, then one host, if it's already infected by one strain, it can still be infected by another strain. So the two strains will just evolve together in the same host. And when they infect another person, one of the strains will infect the other person. Does that make sense? Okay. Cool. Thank you so much.
Okay, welcome back everyone to the second session of the symposium. We're starting off with Ailene MacPherson and the role of tree size and diversification. And I'll hand it over to Ailene.
All right. I'm really glad I got the microphone because I'm a person who likes to walk around. So it's good. All right. So tree size. This is a really central quantity to how we think about biodiversity. Whether or not we use it very often in that way differs, but I wanted to just put up here a set of questions that fundamentally depend on tree size. So everything from why is a clade speciose to why is a clade depauperate, or is it even speciose to begin with, that fundamentally depends on our expectation for how big a clade should be. We could ask other questions like, is diversification density dependent? That's one thing that will come up later in the talk today. And then if we think about lineages as viral sequences, for example, in all the genome sequences we collected during the COVID epidemic, we could ask questions like, how big, oops, well, that went fast, okay. How big should an epidemic be? All right. So I'm going to tell you a story about tree size in 3 different acts. The first act of this is how do we actually figure out how big a tree should be? So we can do this using math, but it is not a new question. It's a question that Yule asked 100 years ago. And so in this classic paper, and he built a model where you only had speciation. It's actually a little bit more nuanced than that. If you want to hear a whole history about what Yule actually was thinking about, check out a forthcoming paper in Phylogenetic by Matt Pinnell and I, where we talk about this paper in detail. But Yule was surprisingly quantitative, and he derived this expression for the expected distribution of tree sizes under his speciation-only model, which later got called the hollow curve, but it's a geometric distribution, so you have lots of small trees and a few very, very large trees. But if we want to do this for more modern diversification models, we're going to need to use more complicated math. And so the first challenge that we have is deciding how to actually measure tree size. What does it mean to count a tree? So I've posed 3 different measures of tree size. The first one is just the number of lineages at the present day, so the extant tree size. The second option that we have is how many samples we've collected. Now, this can be quite different than the number of lineages if, for example, you're sampling fossils or in an epidemiological scenario where you're sampling viral sequences through time. Finally, we have a third measure of tree size, which is how many unique lineages you observe in your tree. And this incorporates an element of topology, because if you sample a fossil and then a subsequent descendant, that doesn't double count. And so this measure of tree size is unique in that it captures both the number of tips in the tree, but some element of topology. But given these 3 measures, how do we develop expectations for how big that tree should be? Well, I'm going to present 2 different methods. The first method is based on a post-traversal master equation approach. So I want to break down this name to help you understand what this method is. First of all, it's post-traversal. That means that we do the calculations from the present day going backwards in time. This is very, very convenient. We've learned as evolutionary biologists that lots of mathematical things are easier calculated backward in time. But it has a disadvantage that it only can see what happens in the future, not what happened in the before the what happened in the past. And so we can't incorporate things like density dependence, because that depends on how many lineages had arisen already and not what happens in the future. So it's going to only work for density independent models, but it's really nice in that it has a natural interpretation. So we can look at this equation, we can say, this is the term for things when nothing happens, this is what happens when we have speciation, here's what happens when we have sampling. So it's very naturally interpreted. A second method we could use is an ensemble moment approximation. Now, let's break down this name. So ensemble moment means that we're going to be looking at the moments of the distribution of tree sizes, not the distribution itself. So just summary statistics of that distribution. Second, it's an approximation. So that doesn't mean we don't get all of the moments. We only get a couple of them. So in this case, I'm going to only look at the mean and the variance. All right, so here's an equation for the mean and the variance in tree size. There are some nice little interpretable bits that you can get out of this equation. You can get, for example, that from the mean, it looks kind of like what we would expect if we were writing down a deterministic model. So the mean should look pretty deterministic. If we look at the variance, there's some changes in signs here, which means that the variance of tree size increases as the number of events increases. So the more speciation events, the more extinction events, the more variable our tree size should be. So that tells us about how we calculate tree size. Now what am I going to do with that tree size? So going back to those questions that I posed at the beginning of this talk, let's visit one of them, which is, does tree size tell us about whether or not speciation and extinction are density dependent? So in order to answer this question, we're going to contrast 2 different models. We're going to contrast an exponential model of diversification. So this is your classic birth-death model to a model with logistic diversification. So speciation slows down as you get more and more species. And so if we look at the distribution of the tree size, we see on the top when we have that exponential birth-death model, we get our classic hollow curve. If we look at the case when you have logistic diversification, it is not at all geometric anymore. It's very normal. And this makes sense, that you have some trees that immediately go extinct. That's that little peak down there at 0. And then everything else just gets really close to the carrying capacity of tree size, so you get a normal distribution of trees. I find it also interesting to look at how the moments change. So in the top, you can see that as the birth rate increases in an exponential model, the trees get bigger. As the death rate increases, the trees get smaller. And if you look at the variance in tree size, it's exactly what we would have expected from that set of equations, that all that matters is the number of events. If you look at what happens in the logistic model, you get a very different pattern. You see this pattern for the mean, and essentially if you have a small tree, you have a highly variable tree. So it's a very contrasting distribution of moments for logistic growth. So does this matter? Well, I want to argue that it does, because there's these wonderful papers showing that it's hard to detect density dependence in diversification. In fact, for every density-dependent diversification model, if you look at branching density alone, there's an exactly equal time-dependent model that is indistinguishable from the density-dependent model. And so how I think about this is that branching density is like looking at a rate. And so if you look at the rate at which diversification happens in a density-dependent model, there's almost always going to be a time-dependent model that has that same rate. But if you look at the integral, so the total accumulation of lineages over time, you can get very different patterns, and that's what tree size tells us. So to test this, we looked at two models, a time-dependent model and a density-dependent model, that look exactly the same in terms of branching density, but we looked at their distribution of tree sizes. And you can see here, notice that the x-axis is very different scale here, that these two distributions are not at all similar to one another. So while we can't see the difference between time-dependent and density-dependent on an instantaneous rate version, we can see those accumulated differences over time in terms of tree size. So tree size does not give us the same information as the branching density. Finally, I want to visit model identifiability. So when can we not infer diversification rates? So I mentioned very briefly this idea of branching density. Here I'm making it more formal. So we usually use branching density in terms of all the times of these speciation events to write down a likelihood for a particular observed tree. And then Luca and Pinnell showed that this branching density alone is not sufficient to pull apart speciation rates and extinction rates. And this is because you only have one piece of information in that branching density when you need to. I'm happy to talk about this more, but the point that I wanted to get to is that there's whole relationship between these congruent models that we can't pull apart is in terms of a differential equation. And this always nagged me. This is really an interesting result, but this differential equation is super opaque. Why? Why are these the same? So I wanted to say, what can tree size tell me about why certain models are unidentifiable? So to answer this question, we looked at the distribution of tree sizes for two models that have exactly the same branching density. So they're congruent in terms of Luca and Pinnell's results. And what we found is that if you ignore extinction, so if you drop those very left-hand 0 bars and re-normalize your distribution of tree sizes, you see exactly the same distribution of tree sizes. This means that congruent models are congruent because they are indistinguishable up to, but not including extinction. So they differ in their extinction probabilities, but not in their non-extinction probabilities. And this makes sense because we don't look at phylogenies that went extinct. So if we only consider the set of phylogenies that didn't go extinct, then we can't tell these two models apart. All right, so I think I'm running out of time, so I'll quickly conclude by the fact that I've talked about how we calculate tree size distributions, that tree size can tell us different things than branching density in terms of density-dependent diversification. It can also give us insights into why different models struggle to be pulled apart, because they only can be differentiated by certain parts of the tree size distribution. And with that, I just wanted to point out that I have a preprint up about this work. I have always openings for grad students in my lab. And then I wanted to have a shout out to our new society for the Society for Modeling and Theory and Population Biology, and everybody is welcome there. So with that, I would like to thank the organizers and take any questions.
Okay, and we're continuing with a similar theme from taxonomic scale importance to phylogenetic scale, and we move from whales to plants. So Tom Carruthers is gonna tell us about that.
Thank you, and thanks for inviting me to having me at the symposium. So flowering plant evolution and phylogenetic scale. So what do I mean by that? I'm essentially referring to the fact that, phylogenetic trees incorporate a massive range of broad to narrow clades. That can be either within one phylogenetic tree or across a lot of different trees. And I think that has relevance to how we use trees to do evolutionary analyses, because the branching events that are represented in those trees and that are defining relationships between the clades in those trees kind of correspond to quite distinct evolutionary phenomena. So I can maybe illustrate that a bit more clearly with 3 examples. So you might have really broad phylogenetic scale, like the whole of flowering plants, and the deep branching events in that trees are defining relationships between big clades like orders, slightly narrower scale, for example, angiosperm order like Ericales. Deeper branching events are defining relationships between different families. Then you might have a much narrower scale like the single genus Ipomea, where the kind of the deeper branching events in that tree are just relationships between different parts of a single genus. And that's quite like a very general overview of what I mean, and I think this phylogenetic scale thing is linked to a whole load of other things, and particularly time. But I hope as I go on through the talk, I'll illustrate a little bit more specifically the implications that I'm kind of referring to and how it's kind of relevant to some of the methods that we use to do evolutionary analyses. So I guess it's kind of a starting point to thinking about that and to kind of moving on from that initial definition is thinking about, does evolution differ between different phylogenetic scales? Now, I guess the argument I would make is that it does, otherwise there wouldn't be that much point in doing this talk. So I think a characteristic you see at really broad scales is that you get this quite rapid emergence of diversity and disparity. You know, a classic example that people have known about and talked about for a long time would be the origin of angiosperms. You know, Darwin talked about the sudden appearance of diverse angiosperm fossils in the Cretaceous, and these 2 diagrams here are just kind of illustrative of that. So on this side here we have the kind of early part of a flowering plant tree of life, and you can see the really rapid branching events giving rise to different lineages. And then the example on this side here, particularly in reference to orders, so it's showing the kind of cumulative origin of orders through time, and you can see that all those orders originate in a really compressed time interval in the Jurassic and the Cretaceous. I mean, there's some uncertainty about exactly when that is, but the point is it's generally pretty compressed. And those patterns aren't just in flowering plants as well, they're kind of observed in association with the origin of major clades at broad phylogenetic scales across the tree of life. And those dynamics, I think, are quite different from what you see at narrower scales where rate increase, I mean, it's hard to define it exactly, but I think rate increases are generally less long, kind of more short-term, and they're often associated, I would say, with less pronounced morphological disparities. Alongside that, I think another key characteristic of broad scale, kind of in the origin of clades at broad phylogenetic scales, is the fact that molecular rate variation and the characteristics of it differ quite substantially across those scales as well. So again, I think quite good evidence of that comes from divergence time estimates. So an example might be the age of flowering plants. So in this example here, you've kind of got a Triassic age for the crown node. This example here, you kind of got a Cretaceous origin time. And those uncertainties at broad clades are, again, not just in flowering plants. There's been a lot of talk of it over the last year in terms of mammals and animals too. And that's relevant to molecular rates, because I think one of the reasons we get those uncertainties is because we're not very good at modeling how rates differ and how rates differ at those really broad scales. And that's not to say that divergence time estimates are good at those much narrower scales and that we're modeling rates really well at those narrower scales, but I think probably the problems are a little bit less profound. Now, I guess the way I've framed it so far is I've kind of talked about it as if evolution happens differently at those different scales, and it's reflecting real differences in the things that happen. I guess it's worth bearing in mind, and a kind of additional sort of complicating factor also is that the information in a phylogenetic tree differs a lot at different scales as well. especially a phylogenetic tree that's primarily made from and estimated from extant samples. So that's illustrated here, where you've got sort of a hypothetical tree of extant and extinct lineages and a corresponding tree that, you know, it's accurate. That's an accurate reflection of that, but it only has the extant taxa, and it would be, you know, equivalent to something you would construct with molecular sequence data. And obviously, as you get to broader and broader scales, you get more and more extinct lineages, and you become increasingly tenuous, really, in an evolutionary sense, about what those deep branching events actually represent, and what their relationship is to evolutionary entities that have existed in the past. So I guess, in spite of all that, you might still ask, why does any of that matter? Aren't we aware of this anyway? And yeah, aren't we aware of this anyway and acting as such? So I guess, in answer to the first question, does any of it matter? I'd say, yeah, it definitely does, and probably more than it ever has done, given that the data sets we're using span ever greater scales, and the way we're trying to do analyses probably is trying to integrate things across different scales. And in terms of the second question, aren't we all aware of it and kind of acting as if we're aware of it anyway? I would say maybe not as much as we should be. The reason I would say that is because I think a lot of the methods we use are quite similar across those different scales. Perhaps a good example, again, would be in divergence time estimation, where we often use similar relaxed clock models across a vast range of different scales. And in general, we use fossil calibrations in pretty similar ways. I mean, I'm not saying we always use them in the same way, but a lot of the methods we use are pretty similar, regardless of the scales we're working at. And we use them to then kind of address questions about, you know, when and in relation to what certain lineages originated, or when they diversified and how they diversified. So two examples would be here. You know, one of them's in relation to angiosperms. One of them's in relation to one angiosperm genus, Saxifraga, and they're both kind of addressing a similar style of question, despite those kind of quite profound differences in scale. So I guess those issues will be even more important if they cause appreciable differences in how methods actually worked. So for the last part of the talk, I want to just briefly summarize some simulations I've been doing that have kind of tried to pick apart this a little bit. They're unfinished, but I hope they can illustrate some of the effects. So they're going to be in the context of divergence time estimation, and in particular fossil the kind of two main components of that, which are how we use fossil calibrations and how we use relaxed clock models. So they're based on, in terms of the fossil calibration part of the simulation, they're based on a kind of a birth death branching process, which is shown here. So you've got extinct lineages, extant lineages, and then fossils are simulated along the branches of that process with the blue circles here. And we're interested essentially in how do different phylogenetic scales affect the expectation of when a fossil originates in relation to when a clade originates. And we're thinking specifically in terms of extant clades, so the ones on this side, because that's kind of relevant to how you would use fossil calibrations to date a molecular phylogenetic tree. So those simulations are done with different amounts of extinction and across different phylogenetic scales. So I will run through a couple of simple results from them. So this first set of results, the graphs basically show the percentage, so run across a lot of simulations, the delay from when a clade originates in comparison to when its first fossil originates. So the fossil is always going to be after when the clade originates, the first fossil for that clade. So in the first example, it's basically just comparing different amounts of extinction, with the kind of premise being that at broader scales you'd expect more extinction. And kind of, I guess the take home from that is that when you have no extinction, you get this quite simple kind of tail off with time about when you'd expect to sample the oldest fossil for a clade, whereas when you start to have extinction or even mass extinction events, the kind of peak of when you would expect to sample the fossil starts to be significantly delayed, compared to when the clade originates, which you would expect would lead to biases in when you estimate divergence times that would be specific to broader phylogenetic scales. Then in a slightly more complex scenario, we've got narrow, medium, and broad scales, and in this case, we're assigning fossils to clades based on simulated characters. So in the first case, I'm assuming we know with certainty the correct extant clade that the fossil belongs to. In this case, the fossils are simulated with a handful of morphological characters, then they're assigned to nodes in the tree based on that. So it's kind of more equivalent to how you would put fossil calibrations in a tree if you're trying to estimate a dated phylogeny. And so you can see in this case, basically the kind of the delay relative to the age of the clade increases as you get to broader phylogenetic scales. This case doesn't have extinctions, so obviously like what would be ideal to do would be to kind of combine the two aspects of those simulations, and you'd probably expect to get an even stronger kind of phylogenetic scale-dependent bias in when the fossils originate in relation to the extant clades. I won't go into that now. That's like an explanation. I can talk about that if anyone wants to, but there's probably not time. I've done a related simulation in terms of implementing relaxed clock models as well. Basically, I'll just very briefly summarize. So simulated phylogenetic trees at different phylogenetic scales and estimated substitution rates at those different scales. The basic take-home is that at narrower scales, you can estimate substitution rates of a higher degree of precision. So again, like with the fossil thing, it's a phylogenetic scale-dependent effect that could potentially, or is likely to cause error in the resulting divergence times. And that's important because divergence times underpin a lot of the other methods that we use. So I guess what could we do? I mean, that's obviously not kind of a complete kind of set of things, and that's for people to decide really. But there's certainly scope for better methods that take this into account more explicitly. I guess just being aware of these issues is useful, both in terms of how we interpret things and in terms of how we design the questions that we address when we're using these methods as well. There's some acknowledgements. So Smith Lab, it's not directly related to what I'm doing with them, but I enjoy working with them, and it's good doing stuff, doing other related things when you're enjoying your work. And then Robert and Tom as well, who I've worked with for quite a few years. So thanks, that's all I've got. So it's time for any questions.
Can I do this?
Yeah, sure. Okay, our next talk is pre-recorded. It's Laura Alencar, but we have a
Hi, I'm Laura Alencar. I am an associate research.
That was rude, Orlando.
I don't know why it started without me telling it to, but
I just wanted to say we have a co-author here that can take questions, possibly, at the end. So if you have questions, and if we have time, you can still get questions at the end.
Where is the mouse?
Hi, I'm Laura Alencar. I am an associate research scientist at the Department of Ecology and Evolutionary Biology at Yale University. And today I'll be presenting the first results of a collaborative project between the Munoz Lab at Yale and the Ueda Lab at the Virginia Tech. So first of all, I would like to thank all my co-authors here. And so I would like to start by showing you some cool pictures of fishes, and by looking at this beautiful phylogeny of coral reef fishes, we can readily note that some lineages accumulate diversity way more rapidly than others, which is here illustrated by the different colors associated with the different branches, where warmer colors represent higher rates. And this is something that we can actually also see across squamates or lizard and snakes, for example. In this case here, the different rates are associated by the different colors in the different dots, where again, warmer colors, higher rates. So these two examples actually illustrate a prevalent pattern in biodiversity. Evolution proceeds unevenly across the tree of life. And one of the major sources generating this diversity disparity is the access to ecological opportunities that these lineages will face during their radiation. So events such as the emergence of a key innovation like the evolution of viviparity or the two pads The invasion of a new environment or the extinction of antagonists such as competitors and predators can create such opportunities and catalyze diversification, because lineage will take advantage of these vacant niches and then are able to generate more and more and more species. However, it would be great if life happened in such a simple, direct, and linear way, or at least would be easier to quantify the cause and effect that these ecological opportunities have in shaping the tree of life. But life is way more complex than that. And what we see instead is a complex network of processes shaping these diversification patterns. So lineage will encounter not one or two, but several sources of ecological opportunities during their radiation, as illustrated here in this hypothetical scenario. And so to isolate the effect of these different sources of ecological opportunities becomes very challenging. But we can do it. And in this study, we use a combination of approaches to untangle this complex network of processes driving diversification rates. So our idea was to first quantify, to first isolate the different sources of ecological opportunities contributing to the rate of heterogeneity, investigate the potential interaction between these sources, and by doing this to come up with more plausible causal scenarios underlying macroevolutionary dynamics. Specifically to do that here, we focus on the diversification dynamics of the Pleurodonta lizards, which represent a large radiation of lizards mostly from the Western Hemisphere. Some of its members, for example, are the famous anoles, the iguanas, the very cute horned lizards, and, for example, the predominantly Andean genus, the Liolimus, which is going to be very important in this talk. And so the first step we took to untangle this complex network of processes shaping the macroevolutionary dynamics of Pleurodonta was what we call the data-driven or the phylogenetic natural history step. So our intention here was to first identify those lineages associated with unique diversification rates, or in other words, those lineages that kind of stand out in their rates of diversification when compared to their background estimates. And so by performing this step, we found several increases in the diversification rates. So here we have a phylogeny for the Pleurodonta, where again, warmer colors, higher rates. And the red circles show the several instances during the radiation of Pleurodonta where the rates significantly increase. And by looking more closely to those lineages with higher rates, we note that many of them appear to share several life history, ecological, and geographical aspects. For example, many of them are viviparous, many of them are arboreal, several of them have large body size or occurring islands. Many of them occur in mountains, and interestingly, many of those that occur in mountains are specifically associated with the South American Andean mountains. And I think it's important to mention that these traits were not chosen at random, right? They are actually widely discussed in the literature as potential source of ecological opportunities driving the macroevolutionary dynamics not only in lizards, but in several other study systems. And so, okay, after identifying this aspect as potential source of ecological opportunities, we wanted to explicitly test for potential correlations between each of these traits and the diversification rates. And to do that, we performed several phylogenetic correlations, which suggested that reproductive mode and Andean mountains were the only two traits significantly associated with the diversification rates. Specifically, viviparous lineages and or those that occur in the Andean mountains have higher diversification rates. But we knew that in Pleurodonta, many of those viviparous lizards also occur in the Andean mountains. So this led us to wonder, are these two aspects, two independent sources of ecological opportunities, independently contributing to this rate heterogeneity, or are they maybe confounding each other in doing so? So to find that out, we took a different approach by using a combination of SSE or state-dependent speciation extinction models. And the first method we use is called MISI, what basically MISI does is to quantify the number and location of the rate categories that best explain the diversification rate heterogeneity across a given group. Specifically in Pleurodonta, MISI suggests that 4 rate categories best explain their rate heterogeneity, which are here. These rate categories are illustrated by the different colors. And just to give a brief idea of who belongs to each of these categories, the first category comprises all the species of the genus Liolimus, those predominantly Andean genus that I mentioned in the beginning of the talk. The second category comprises all the species of the genus Fimaturus, which is also predominantly Andean, and together with Liolimus comprise the family Liolemid. The third category is a combination of the Scalopus subclade and iguanas. And the fourth category is comprised the remaining lineages in what we call the background rate category. And so the next step, we wanted to compare the rates between oviparous and viviparous species and those present and absent in the Andean Mountains, but now within each of those rate categories detected by MISI. So just to make this box plot a little bit clear, each pair of box plot here represent one of these rate comparisons between within each of these rate categories. So in the first set of analysis where we compare the rates between lineages that are absent and present in the Andean Mountains, this analysis suggested that the presence in the Andean Mountain has a positive effect both in the Phymaturus and in Liolimus rate categories. However, when we did the same thing, now comparing rates between oviparous and viviparous species, it seems that viviparity only slightly increased the rates in Liolimus and in Liolimus only. Just to make this a little bit clear, in case you're wondering, but I can see that viviparity seems to increase the rates in Phymaturus. All Phymaturus species are actually viviparous, so we can't make this comparison here. What you're actually showing is just an effect of the prior, not the empirical estimates. And so, okay, to further tease apart the role of reproductive mode and the presence in the Andean Mountains in driving the rate of heterogeneity in Plerodonta, we now combine these two traits in the same analytical framework using the best clade to do that, which is the Liolimus. And what do I mean when I say that Liolimus is the best clade to do this? So remember that the previous analysis, MISI analysis, suggested that all Liolimus species belonged to the same rate category, which kind of help us to control for potential hidden states in driving those rates. Also, Liolimus is actually the only clade in Plerodonta comprising species that are either viviparous or oviparous, and also they're comprising species that are absent or present in the Andean Mountains. So it's basically the only clade that we can do this analysis. And on top of that, it is one of the clades in Plerodonta with the higher diversification rates, which in my opinion just make them even more interesting. And so we found that in Liolimus, those viviparous taxa that are present in the Andean Mountains have higher diversification rates than those viviparous taxa that are not in the Andean Mountains. And so this led us to conclude that viviparity has only a minor effect in driving these diversification rates, and it is the occurrence in the Andean Mountains, the factor, the trait that is actually increasing these rates. And so this first set of results show to us that some lineage-wide signatures, like for example viviparity potentially driving the diversification rates, are actually lost when we analyze subclades, like when we did with Liolimus. But what about the opposite scenarios? What are the odds that we are missing signatures of traits in driving rates because we're focusing on broad phylogenetic scales? So this came to our mind specifically when we're thinking about body size. Because body size is a relative metric, right, to the geographic, phylogenetic, or ecological context that an organism belongs. So a crown giant anole, for example, is not that gigantic compared to another arboreal species, the green iguana. The arboreal green iguana is way larger than a viviparous terrestrial lizard like the Phymaturus, but this terrestrial viviparous Phymaturus is actually way larger than another terrestrial viviparous lineage, the Liolimus pictus, illustrated here. And so remember that we didn't find any association between body size and diversification rates when we analyzed all the Plerodonta species together. However, when we analyze and compare the rates within each source of ecological opportunities and also combining different taxonomic scales, we do observe that larger species, here represented by the brown triangles, have higher diversification rates. And so the second set of results suggests that some signals fade at broader phylogenetic scales and become clear, but they become clear at shallower ones. And so our findings here, I think, bring both or more organismal focus and also methods-oriented conclusions. So first, viviparity, while viviparity appears to accelerate diversification, its effects is clearly overestimated by the association between viviparity with the Andean Mountains in Plerodonta. Also, Andean colonization drives Plerodonta diversity, but it's important to acknowledge that Liolimus, and I would add Phymaturus here too, they have their own unique dynamics. There are also other things that we're not addressing here that contribute to their super high diversification rates. Importantly, while some sorts of ecological opportunities are sufficient on their own to boost diversification, others, such as, for example, body size, must act in concert with additional factors to achieve a similar effect. And specifically addressing the topic of this symposium, We highlight that while phylogenetic comparative method offer valuable insights, they may be also influenced by the complex interaction of factors underlying rate heterogeneity, which potentially leads to like false positive and false negatives as illustrated here. And so to help overcome these methodological challenges, methods must embrace this complexity that characterize biological systems, and quantifying the effect of latent factors on diversification is definitely a way forward. And finally, it's important to mention that we are limited to the traits that we are analyzing, which represents only a limited snapshot of an organism's biology, right? So we can only test as much as we know about the organism. So by combining phylogenetic comparative methods with natural history, we can identify more plausible ecological opportunities in these large evolutionary radiations. And I would like to conclude by saying that making progress in natural history is also making progress in macroevolution. And so if you're interested, this study was published a few weeks ago in Evolution Letters. Feel free to download this open access. I would love to have some feedback. I also know that I walked through a lot of methods here, but we put those in a step-by-step fashion in the paper. So we hope folks are able to use this framework in their own study systems. And finally, I would like to thank the symposium conference organizers for giving me this opportunity to be there without being there. Thank you all also for being here today, and feel free to email me if you have any questions. Thank you so much.
Well, we're coming to the last talk of this session. Gustavo Burin will be talking to us about mutualism breakdowns in time and space. I think, again, we have some time, but it's the last one in the session, so.
Hello, everyone. Thanks for coming. Thanks for sticking by on the very last talk of the day. It's hard to compete with lunchtime, so I really appreciate you being here. I'm Gustavo. I'm currently a postdoc at Durham University at Guillaume Choumiki's lab. First, shout out to whoever takes the time and space reference there. And I'll be talking a little bit about the project that I started about 3 months ago. Unfortunately, I have a sad disclaimer. There won't be any results today. But by the end of my talk, I hope I can get you as excited and maybe scared as I am of working at these scales. Initially, I want to thank Mari and Orlando for putting together such an amazing symposium, and also all the people that are involved in this project. I'm like a stellar group of people. I'm learning so much from them, so it's a real pleasure to work alongside of them. It is not a novelty for anyone in this room, I think, that the flowers and plants, flowering plants and seed plants are very diverse and have accumulated an immense diversity over geological timescales, right? And this amazing diversity is both a consequence and a cause for many different types of interactions with other species. In this particular case, I'm highlighting a few mutualistic interactions, such as seed dispersal, pollination, nitrogen and nutrient fixing, and also interactions with plants for defense of those, with ants for defense of those plants. And also, as a cause and a consequence of those interactions, many co-evolutionary processes have acted, and both shaping and being shaped by this diversity of plants over time in multiple different groups, such as vertebrates and different groups of vertebrates over time. So it's very important for us to understand how these different mutualistic interactions evolved over time, right? So this is a quick example of different pollination modes which were inferred back in time to see and to estimate which was the ancestral state for this for pollination, for this mutualistic interaction. But I think almost probably as important as knowing how these mutualistic interactions evolved, it is very important to understand how they break down across the tree of life, of the plant tree of life. So we can see there are many changes between abiotic and biotic types of seed dispersal in this case in particular. So I think it's also very important to understand when, why, and where they break down. So the major goal of this project is to understand a little bit of when, where, why, and how these mutualisms break down across the plant tree of life. And how are we going to do that? So I'll give you a brief overview of what we have in terms of data right now. we're dealing with many different types of mutualistic interactions. So as I said already, pollination, seed dispersal, mycorrhiza interactions, and nitrogen fixing bacteria, also ant-plant interactions. And we have assembled, and we are still finishing assembling an amazing data set for each of those types of interactions. I'm putting you here some figures for you to have a sense of the scale that we are working on. So for pollination, we are working with around 31,000 species, and it's the same number of species for seed dispersal, but we are still finishing filling out the gaps in this data set. We have also information in different levels of organization for mycorrhizae interaction, for instance. We have expert information for virtually all the genera, but also we have direct information for almost 15,000 species. And for ant-plant interactions, we also have information for all the species for which those traits were ever reported. But not only that, like these numbers are already amazing, and it will be hard to deal with. But we have an ongoing collaboration with Dr. Rensky-Onstein from Naturalis. And in addition to that, also the TRI database, for which if we combine the three sources of information, we might reach 57,000 species with seed dispersal information. So it's a lot. It's a lot. And to do that on a phylogenetic perspective, we will use the most complete tree possible, which is based on the Smith and Brown tree from back in 2018, which had around 80,000 species. But with the recently published phylogenomic tree that came out this year, Stephen and his group are already working on incorporating new sequences that were deposited in GeneBank in between these two publications. And we heard back from him, and we know that now the new tree will have at least 134 species, 1,000 species, which is also a lot. So that's why I thought about being scared about this type of data. So just to give you a brief overview of how these, the species for which we have traits are distributed across the tree, this is still the topology from 2018. This will change in the near future. But the red tips here show species for which we have information either for seed dispersal or pollination, which is nice to see because they are quite evenly spread across the tree. So this prevents us from suffering from some major methodological issues. But also we can, if we zoom in into some groups, we start to see that we have many different patterns of evolution of different traits. So for instance, for the Asparagales orders, we can see that the bottom phylogeny represents the pollination information, and the top represents seed dispersal information. So if you see the color of the small dots here, for Asparagales, both traits are kind of highly conserved. There's not much variation in each of those traits. But we also have cases in which one trait is very conserved, for instance, pollination in Asterales, the daisies and sunflowers. But we have quite a lot of variation in terms of seed dispersal for this order. And lastly, we also have cases in which both traits can vary quite a lot. So this brings interesting questions that we want to address in the future, for instance, like correlated evolution and other things like that. And at this scale, it will be basically a methodological nightmare to work with all this data. So it will take quite a lot of computational power. And also, I'm already anticipating that it will take quite a long time to run those analyses. So yeah, hopefully I can finish those analyses by the end of my contract, but let's see, to be defined. So one approach that is possible to address the questions that we want to address using such a large data set is an approach that I used to call a divide and conquer. So instead of looking at the whole tree, we can take a look at smaller clades. In this case, I'm representing three of the three large orders that I showed you before, three of the orders that are mostly represented in our data set. And this could be our units of work. So instead of looking at the whole tree, we can take a look at major clades and see how they behave, acting as like phylogenetic replicas. And we can do this either by hand and arbitrarily, by taxonomy, or by knowledge of the natural history. But we can also use some algorithms that were published recently to have a first scan across the tree to identify possible places where shifts might have happened and look into that into more detail. But ideally, we would look at the whole tree altogether. So, but it's not unsurprising that there should be a lot of rate heterogeneity across the tree, especially a tree this big. So, and usually discrete trait models, they are fairly simple, like the MK models tend to be very simple, so it is important to incorporate this heterogeneity in any way possible. One way would be to use hidden rate Markov models that could not only map the estimate rates for the explicitly mapped trait, but also try to understand what is going on within each of these categories through hidden rates. So which would allow us to incorporate this rate heterogeneity that I told you about. The package also includes some correlated evolution models, and that allows us not only to test like pairwise, but also include more traits evolving together. But we are mostly interested in the possibility to obtain tip-specific rates. And I will tell you in a bit why are we interested in those tip-specific rates. And very recently, this year, like a few weeks ago, Luke and Liam published this preprint where they propose a new model, gamma-distributed rate heterogeneity, that will allow us to not only get the tip-specific rates, but also branch-specific rates, like the internal branches would also have their own rates, which would be amazing for us to trace how these traits evolved through time. Going back to how they are distributed, the numbers are quite impressive. The number of species, like 57,000 species, is a lot. But still, dealing with plants, this represents a minority of the species. So there's still a lot of missing data and lots of species without any trait information. So this is yet another thing that we will have to circumvent and to probably run some sensitivity analysis and so on, so we can properly understand what is going on in our results. So if, but more optimistically, when we overcome these challenges, the main goal of the project would be to trace the evolution of mutualisms and the mutualism breakdowns across the plant tree of life, not only on time, like this example, but by having branch-specific or maybe tip-specific rates, we would be able to also plot them on space to maybe see if there are some hot spots or cold spots of breakdowns in these different types of mutualistic interactions. So the take-home messages, I hope I have convinced you that we have an amazing data set and amazing phylogeny. I'm still thinking about the approach, so I would like a lot to talk to whoever is interested in that, discussing different approaches, so the divide and conquer, or a more integrative one, how to deal with the missing species. But I think not only conceptually and biologically we will have a lot to add with this project, but we will also be able to provide some methodological advancements, such as new approaches or maybe faster implementations of current approaches, which will be key for us to finish this project in our lifetimes. So with that, I would like to thank you all once again, and I would gladly take any questions. Yeah, no, so that's basically the idea. If we have, because the way we are building the data set is based on the species list from Amy's phylogeny from back in 2014, but also based on Smith and Brown's phylogeny from 2018. So that's, I understand that the phylogenies at lower organization levels should be more precise and more robust, but then we risk losing this association between our data set and the phylogenies. But the whole idea is that the downside of it is that at the root of each of those groups, you cannot inform the method how the sister groups behave. So my idea is to maybe come up with some way of doing like iterations between different parts of the tree to speed up the process, but this is still to be developed. Okay. Thank you once again.